My name is Herbert Saro. I'm at the Department of Bioengineering at the University of Washington in Seattle. In this short video, we're going to talk a little bit about our new paper. It's called Design Patterns of Biological Cells, and it's a number of us have contributed to this, myself, Steve Andrews, and Steve Wiley. I'm sure many of you recognize this textbook. It's the Albert's book on molecular biology of the cell. It's 1,500 pages. It describes in detail many of the molecular mechanisms we find in cells. But it's a huge amount of work, difficult for any one person to absorb. And we started thinking, is there any way we can abstract this information at a higher level to help us comprehend what's going on in a cell? And we were struck by this other book, which is totally different, Design Patterns. It's a famous book in computer science by the so-called Gang of Four. It's an interesting book because it describes how to solve complex problems in a sort of simplified way. And we thought, is there any way to marry this book with the Albert's book? And that's basically what motivated us to write this paper. So the Design Pattern book, which we took our cue from, splits descriptions of systems to three types: creational, structural, and behavioral. So we did the same. To give an example, creational might be the synthesis of protein. A structural might be a cascade, phosphorylation cascade, with an input and output. A behavioral might be an oscillatory system. To give you an example of what we did, this is a sheet I've got which contains on the right-hand side the design pattern, and on the left-hand side an actual mechanistic implementation. So if you look at the top three, the first three, you can see that they are quite different, but the design pattern is the same. Now these examples are oscillators, they're specifically relaxation oscillators, and the actual mechanisms are quite different, but the design pattern is the same. So this allows us to describe an oscillator in a much higher level than we would if we just looked at the mechanism. So we do this for many, many different systems in the cell. And the paper covers a wide range of systems. So we think this paper is important for a number of reasons. One, it's a new way of communicating ideas in molecular biology. We have on one side computationalists and theorists, and on another side experimentalists, and often they speak different languages. This way of looking at biology will allow people to talk about the same thing in a common way. The other idea that we think might be useful here is it forms a scaffolding on which to build further patterns. Now the paper, although it describes many patterns, it's limited. And especially in the behavioral category, there are probably many more patterns we could imagine. For the next steps, we're thinking of a number of things. Probably the most important is to build a database. At the moment, the pattern paper is literally just a paper, but it would be really nice if we could put these patterns into a database that people could search. For example, a given pattern is the relaxation oscillator. And attached to that, we could have many possible implementations of that relaxation oscillator we find in nature. The other thing we'd like to do is, if the database is big enough, we could start applying machine learning to allow machine learning to recognize the patterns in new networks that are found in nature. This is particularly the case in signaling networks, where it's very difficult to understand what function is actually being carried out. If we could have a machine learning algorithm that could inspect a signaling network and perhaps distill out of it patterns it finds, this could really greatly increase our ability to understand what these networks are doing. So we'd like to encourage everyone to read this paper. It's written in a very clear way. Steve Andrews, who is the main author, did a fantastic job in writing this paper in a very readable form. It covers a wide range of biology, and it should interest people from all walks of biology.